My name is Bethel.
I'm Christina.
And we are students at the Governor's School at Innovation Park, and we'll be presenting biofilm and its eradication using proteolytic enzymes.
Biofilms are structures that offer an optimal protective shelter for bacteria to share nutrients, securely develop, and adapt to their environments, consequently causing persistent infections that are rarely resolved by the immune system and additionally respond inconsistently to antimicrobial treatments. A model example of biofilm-related infectious diseases is S. epidermidis, as it is the leading cause of device-related bloodstream infections, which end up costing hospitals across the U.S. approximately $2 billion per year. Here, it is anticipated that a greater understanding of biofilm properties should lead to a novel or modified effective method for biofilm control, and therefore an improvement in the safety of the millions of patients that receive implanted medical devices across the globe each year. That being said, the aim of this study is to determine whether significant degradation of the biofilm matrix occurs when exposed to select commercial proteolytic enzymes, which determine the extent to which S. epidermidis' adherence is affected by surface wettability. We hypothesize that if S. epidermidis was exposed to alkylase or amylase, then its amount of biofilm degradation will increase, causing its adherences to surfaces to decrease, and that its biofilm will adhere more to hydrophilic surfaces due to its not fully yet understood surface proteins. Our independent variable was the alkylase and amylase. Our dependent variable was the amount of adherence and degradation of the biofilms, and the positive and negative control were 10% bleached and the hydrophilic and hydrophobic surfaces respectively.
12 stainless steel pieces were cut and 12 carnauba pieces of the same dimensions were made using a mold and placed into 24 separate tubes. The bacteria was then rehydrated and incubated for 48 hours. Afterwards, 20 milliliters of Luria broth and 200 microliters of rehydrated bacteria were placed into the tubes and incubated for 48 hours. Then 250 microliters of amylase, alkylase, and bleach were added to their respective tubes as shown in the diagram below, with three replicates for each substance on both the hydrophilic and hydrophobic surfaces. After 48 hours at room temperature, the tubes were vortexed for 30 seconds, centrifuged for 5 minutes, and vortexed again.
We next vortexed the tubes for 30 seconds, centrifuged for 5 minutes, and vortexed again. Then we pipetted the biofilms from the tubes into the 96 well plates and added 125 microliters of a 0.1 crystal violet solution in each well, and then we let it sit for 24 to 48 hours. We then rinsed the plates with distilled water and placed under the Synergy LX for a light absorbance method.
For data analysis, the graph represents the light absorbance of S. epidermidis biofilm on hydrophilic and hydrophobic surfaces. A low light absorbance value indicates that light was able to pass through easily due to effective biofilm removal. Results proved hypothesis correct, as S. epidermidis adhered more to hydrophilic stainless steel, and alkylase was found to have degraded 10.2% more biofilm in the hydrophilic group and 60% more in the hydrophobic group when compared to the negative control. A two-tailed t-test was conducted between the control and experimental groups, finding the results to be not statistically significant.
We plan to reconduct and consolidate experiment results using the papain enzyme similar to a dipeptidase to target the extrapolymerase substance and test its effectiveness compared to amylase and alkylase. We also want to test whether a cocktail of these enzymes enhance degradation on the effect of the biofilm.